pessoal, boa tarde. Olá, boa tarde a todos. Eu sou Tatiana Sarruan, chefe de gabinete de Bio-Manguinhos. Em nome da diretoria, mais uma vez gostaria de agradecer a participação e a presença de todos. Hoje somos mais de 800 participantes, tanto presencialmente quanto virtualmente. Muito obrigada pela presença. Como já foi dito aqui pelo Dr. Akira, pelo Mário, nosso presidente, e Maurício Zuma, nosso diretor, a Fiocruz está em solidariedade com a situação.
Fiocruz is concerned about the situation in Rio Grande do Sul. On Friday, we had a situation room that was set up for emergencies in support of the Ministry of Health. They're designing strategic actions to face this acute crisis that the government is facing. And I would like them to share with you and to ask all of you so that we'll be united for Rio Grande do Sul, and we'll be putting together resources from the civil society, and you can use the QR code so that these resources will be immediately used for water, hygiene kits, and other basic inputs. All of these resources obtained will be administered, will be managed in an exclusive account of the emergency fund of Fiocruz and directed wholesale to the victims of the strong floods. And at the end of the foodstuffs, basic foods, personal hygiene items, cleaning items, food for pets, and water. Thank you so much, and have a good event. Let's cooperate. It's good for the soul and the heart and for everyone. Now, continuing the program, we would like to invite the chair of the next panel, Accelerated Biotech Development, key points that have led Korea and China to become He has a PhD in pharmaceutical sciences from the State University of Maringá, has a master's in toxicology, applied toxicology, and medical surveillance from the University of Londrina, and an expert in sanitary surveillance by Fiocruz. He has acted as a scientist and researcher of the International Vaccine Institute and as a specialist in regulation and sanitary surveillance at Anvisa. As the lecturers of this panel, I'd like to invite the director of the European Regional Office of the International Institute of Vaccines, Christiane Gerke, who will make a presentation on the Korean approach study case. She has a PhD in immunology and host pathogen interactions from Stanford University in the United States, and a doctor in microbial genetics from the University of Tübingen in Germany. He's got experience in translation of science and vaccines, innovative candidate vaccines, all the way to phase 2 clinical essays and/or partnerships with a focus on global health and preparedness for pandemics. He has acted on the Institute for Vaccines and Global Health at Novartis in Siena in Italy. And he was the head of the program of vaccines and co-leader of the recently created initiative of vaccinology and immunotherapy at the Pasteur Institute in Paris in France. As online lecturer, I'd like to invite the academic member of the Academy of Sciences, George Foucault, who will make a presentation, the study case, Chinese approach and study case, that's his presentation. And he did original discoveries, significant discoveries on the mechanisms of pathogens and the interaction pathogens, hosts, of infectious hosts, emerging infectious hosts. The interests include global public health and the global health strategy. He is an advocate of international cooperation in public health. Global public health is a pioneer in research and control of COVID-19 after the discovery of viruses and development of vaccines and monoclonal therapeutic strategies. Is a member of the National Academy of Sciences in the United States, is a foreign member of the Royal Society of the United States, and a member of the German Academy of Sciences. Leopoldina is a professor of the Institute of Microbiology and a professor of the Center Control and Prevention of Diseases, and a member of the World Health Organization, expert of the World Health Organization and the WHO One Health. Gustavo Mendes, please, if you can come to the floor. Good afternoon, everyone. This is an international session to integrate our speakers. I will make an introduction in English. Those of you who are listening to translation, please wear your headsets.
This session has the intention to be an international sharing experience, especially focused in Eastern countries which have excellence in biotechnology. This is an important point to highlight because Korea and China are countries that come from experience of having limited number of technologies, biotechnology specifically, from some time ago, and now they have global and strategic leadership in the market all over the world. It's important to highlight this also considering the fact that Korea just recently, the KFDA, which is the Korean regulatory agency, has just in 2022 achieved the maturity level number 4, which is also an important stakeholder when we think about biotechnology development. And China, and I remember since I was working in Anvisa, working in ICH. ICH is this international forum that gathers regulatory agencies and companies to harmonize requirements for quality, safety, and efficacy of products, which include vaccine and biological products. I remember that China, in the beginning of 2016, that was when Brazil started, and Anvisa more specifically, started to reach international standards for regulatory actions. China at that moment, the Chinese regulatory agency was starting what we can call revolution in the way they do regulation. And this is something that is impressive to see, and when you look the details of history, impressive to see that now the Chinese regulatory agency, which has improved in the way they are doing regulation and the way they are dealing with biotechnology, has reached standards, ICH level standards, compared with the major countries in the world of biotechnology. So this session will be important for us to see, to watch, and learn, because we do not have to reinvent the wheel. We use this also here. Reinvent the wheel is a position where we have to do all over from scratch, all over again. We can learn a lot from these countries, and we are expecting these panelists to share with us some of the history that we could look and we could see what we can try to use in our experience here in Brazil. So thank you for accepting this opportunity. And I'll start to ask Chris to come and give the talk about the Korean experience. Thank you.
Thank you. Hello? Does it work? Yeah. Boa tarde. E obrigada aos organizadores pelo convite. And now I switch to English because that's all.
Eu
The pointer. So you might be wondering why someone from the International Vaccine Institute is standing here to discuss biotech development in Korea. And that's why I'm giving you first a little bit of an introduction about IVI. So IVI is an international organization. It was founded in 1997 based on the action of the United Nations development programs, and it's actually headquartered in Korea. So you start seeing the link why IVI would discuss Korean biotech. And they have worked with the Korean government, with Korean biotech for a very long time, well, basically all the time they existed. So the mission of IVI is actually to discover, develop, and deliver vaccines for the most vulnerable populations in the world, which are basically the people in low and middle income countries. Of course, there are also vulnerable population in high income countries, but the focus is really on low and middle income countries. And basically, we work on what we call the unincentivized vaccines. So vaccines where there will be no big margin or blockbuster to be made, and typically the big pharma in the West is not interested in. So where are we all located? Oops, that's a challenge. So as I mentioned, headquarters is in Korea, and it's just very recently, actually last year, that the office, now there's a regional office in Europe, a country office even in Austria, a global health institute in Hong Kong, and there will be 2 more institutes located in Africa, in Rwanda and Kenya, starting this year, and there are still 3 collaborating centers also in Africa. So as I mentioned, IVI is an international organization, so it's not a company, it's not really public. We have member states, as you see here, and there are 44 member states as of now, and the funding actually works that some of the member states, notably Korea, Sweden, Finland, and India actually pay a little bit to IVI, but most of the funding is grant-based. So that's where most of the funding comes from. And here you see, oh sorry, this pointer is weird. So here you see in a bit more detail how IVI does it. So Discover is basically all the basic research. It can be in-house development, it can helping others. There are labs in Korea. They do everything from assay development, new strains, vaccine development, process development in the science department. The develop department is mostly clinical trials, regulatory help, contacts with manufacturers, but you might see that there's actually no manufacturing in the IVI activities. So if we develop vaccines, they are all licensed to commercialization partners or production partners. Delivery here is mostly when the vaccine is already licensed for post-marketing licensure, follow-up, maybe if still needed WHO qualification, and rather at the beginning of the whole pipeline is the epidemiological surveillance to see which vaccines are the most important. So basically, IVI does vaccine development from end to end. But I'm not here to discuss IVI. So let's talk about Korean biotech. And here, when I looked at my slides and was yesterday in the panel for the challenges or how can we jumpstart the Brazilian biotech, I think it was Dr. Akira who said, well, now with the COVID vaccine, there's an increased discussion how Brazil can become self-sufficient. Korea had that discussion with the pandemic flu in 2009. Because they had some internal flu vaccine production, but for the pandemic vaccine, it was not meant to be internal. They tried to purchase the vaccine. They actually committed to purchase from the US, but the US had a rule that if they need the vaccine they produce within the country, they can cancel your contract. So Korea had paid for it, but at the end they couldn't get the vaccine. So this really sparked the discussion, how can we avoid this in the future? And basically that came with the very ambitious aims to become self-sufficient for vaccine production. At that time, it was 2010, they were 20% self-sufficient. The target is 80%, so quite a jump. And they also want to become vaccine manufacturing country number 5 in terms of volume economy. At the time, they were number 11. So just to complete the flu story, it's eventually, of course, they got vaccine for the pandemic influenza, but then also the Ministry of Health established what's called a transgovernmental enterprise of pandemic influenza in Korea. There was the first spark of funding, 65 million in this case, and it has resulted already in the approval of a cell-based vaccine from SK Bioscience. But leaving flu, more in general, it was really the year 2010 that is sort of the start of a new growth of biotech in Korea. And you see here major things that actually, came together. So government investment virtually doubled, private sector investment tripled, and patents also doubled. If you wonder about the conversion, it's, I would say it's roughly about 1,000, the easiest. So 1,000 won come to $1. It's a little bit less in dollars, but for the approximation, close enough. So this slide really highlights a lot of the actions that came together. So government had really identified biotech as a future technology. And then the Korean government is quite pragmatic. Once they decide, they think about the money and they execute. So there was money flowing in, as you see in the next slides. It was also a lot about innovation. So if you have new innovation, you can create new things, as was discussed this morning in the panel. It's not just recreating what others have done. So there are more opportunities for growth. And right now, for example, Korea is one of the most developed technologies for patches, like the ones you put on the skin. They use it in cosmetics, not yet in vaccines. Some big companies are not interested, but maybe some of the small companies in Korea could move it forward. Then a lot of large Korean companies actually picked the biotechnology as a sector also in around 2010. So for example, when I hear Samsung, I think about telephones, but not about biotechnology. But they have actually become one of the largest CDMOs in Korea to produce, whatever you ask them to produce. SK, the same. It was in energy at that time. They started SK Chemicals, and then later on it became, or it was a spin-off to SK Bioscience. New companies were founded. For example, U Biologics. Dr. Parks was here on the panel for the DVMM manufacturers yesterday. I mean, U Biologics started in 2010. Actually, the oral cholera vaccine from IVI was the first project they worked with. Now they supply 90% of the world's supply of the cholera vaccine, and they've also picked up other products. So there was really a conversion between the IVI interest and U Biologics interest to help them grow. But then a lot of additional things happened in Korea. So in 2000, so basically they started discussing with companies, thinking what is really needed, what do we do? And the first concrete action now for vaccine specific was in 2016, the action what is now called Vaccine 3.0, which was basically a $200,000 investment to boost, to build the first CMOs. And I'll explain a bit more on the next slides. In 2018, the Right Fund was created. So as you heard this morning about the GHIT, the Right Fund is a combination of funding from the Korean government, which is about 50%, 25% funding from the Gates Foundation, 25% funding from Korean government, from Korean companies. And from the time it was started in 2018, by now the volume has doubled. And the aim of the Right Fund was really to basically address the first valley of death we discussed this morning. Academia has a lot of nice ideas, and then no funds or no means or no knowledge to move forward. And the Right Fund hits exactly there, to take the projects from the academia, help them to move forward to companies to get it produced, to at least move to a phase 1 trial. The Vital then was a later initiative in Korea in 2020. It was another $200,000 investment, more focused on clinical trials. And now we are talking even about the K vaccine hub, which is a $2 billion investment. So you can already see that some of the factor in Korea was definitely money coming from the government. On the very bottom here, you see the self-sufficiency of Korea in vaccines. As mentioned, it was about 20% when all the action started. In 22, it was maybe around 40, sometimes 50% is not quite defined. And the goal to reach 80% now looks like maybe 2030. And these were just the different partners IVI has worked on the way. But to go a bit more into detail on the initiatives. So here's the Vaccine 3.0 from 2016. That was really to support the vaccine R&D. So they built actually a research center with some, really for research and vaccine development, which is the one part. And then there were 2 CMOs exclusively for vaccines, one based on animal cell culture, so you would grow viral vectors, etc. One for microbiology products, so if you want to make killed bacteria, life attenuated bacteria, similar. And it was actually the world's first public CMO, and production size was a bit beyond the phase 1. I mean, it could go up to 1,000 liters and even 200 liters in the BSL-3 production system. So this is what then allowed some of the small companies to go and have their vaccines produced here and continue to clinical trial development. Then for the VITAL, so this is the Vaccine Innovative Technology Alliance, was funded in 2020. And here the mission really came back again to the vaccine self-sovereignty. And they now thought about what all needs to be strengthened to actually get it. So with the Vaccine 3.0, they had built the first CMOs. Here now the goal was to focus more on the clinical conduct, clinical trial development, regulatory development, as Gustavo already referred to, but also training. Because you have to think, if you want to do all of this, you also need PhDs, you need technicians, you need people who know about vaccines. And that's a big part of the aim here. So here again, then the aim to become among the big 5 was again reiterated. So you see the period is aimed at 2020 to 2029. Here there are about $200 million to complete 7 vaccine trials, including 3 for the National Immunization Program. And here the target was still really the National Immunization Program, so the childhood vaccines, not any pandemic vaccines, to look at that and also other global unmet needs for vaccines or new technologies. So here it's a bit more in detail about the goals for example, in the first case, the self-sufficiency. Here the status was, it started at 40%, it should go to 80%. And the vaccines they wanted to boost here is Japanese encephalitis, the DTP, hepatitis, and HPV. So you see it's not any COVID, MERS, SARS, etc. These are really the vaccines that were needed in the country. Of course, more on the researchy side, they were also looking at a lot of vaccines for the global unmet need, and then thinking about vaccine technologies, what's really needed. But the big boost now comes, at least financially, with the K Vaccine Hub. But suddenly you also see a transition of the priorities. So this was born by the COVID pandemic. And suddenly the ambition was not only to become self-sufficient, but actually to export. So now there's much more goal exporting vaccines. And the 2 billion will actually build the manufacturing facilities, or aim to build the manufacturing facilities that will be required for that. So to look a little bit at the shift, so you see here the vaccine self-sufficiency. If you look at the scale on the bottom, you see that are sort of the old vaccines, of which many are the childhood vaccines. And the VITAL, you see, was still targeting really those vaccines. But now with the COVID pandemic, there's the aim to get new technologies to Korea or to develop new technologies, to really become a global leader, not only for the currently pandemic vaccines, but even to address future pandemics. So there's a bit of a shift away from the self-sufficiency to the future pandemics. Sorry. And this is only an example how it looks now. So for COVID, Korea was not yet aiming at being self-sufficient. It was actually the plan to buy. But at the same time, they also developed vaccines which now can be used as booster. And for example, the one from SK Bioscience is now approved Others are in phase 3 trials. Some have stopped. But it's now a very rich landscape. Many institutions have tried to develop. And it's just a sign of that the whole principle of innovation and trying to build is working, even if for this pandemic, or the last one, luckily, it was still, let's say, too early and also not planned. A completely different aspect around all of this is really training. So you might be aware that WHO actually had a call for what they call a global biomanufacturing training hub. And several countries applied, and Korea actually got the go. So there's a lot of emphasis right now. The first iterations have started. There's a, let's say, more like a theoretical course, which is the module 1. Anyway, you will find it. So that started last year, and it will build and build. So first it's a theoretical course. Let's say it gives in 2 weeks a whole overview of vaccine development from research through preclinical, what's all needed, toxicology studies, clinical trials, what's needed for licensure. So it's a big package for 2 weeks. But then it's followed up with courses more on the GXP training. So those 2 exist already. It's still a bit more theoretical, but then it will go into hands-on training, more modules to arrive. You see here, there's the last one will be about entrepreneur training, which was also a topic this morning, that just because you have the technology, you will still not know how to go forward. So it's a big effort, really. So at the moment, it's still on the introductory and the GMP. These are the ones that have been implemented, and then you see the intensive course. And at the end, it should be a master degree course, and actually what it's called the Global Biocampus Program. So here are just a few impressions how it looks like. The courses at the moment is between case studies, lectures, mini conventions. Sorry, I cannot read it there. Visits to companies, and of course here the GXP experience. And this is a summary basically of the slide before, showing you where it all goes. And this is already it, what I have to say. I hope you can find some useful information in it, and I look forward to the discussion. I want to thank Mangi Song, who is the Deputy Director General of Science at IVI. I mean, he has been in IVI, I think, about 20 years, so he has worked with the Korean government and the Korean industry really from day one. And normally he would give this presentation and not me. So he gave me a lot of his slides, and so thanks to him. And then the partners in Korea, the funding partners, without whom the work also wouldn't have been possible, because, for example, the first project with U-Biologics was funded by the Gates Foundation. And then all the other partners IVI works with, and of course the colleagues at IVI. Thank you.
Now we are ready to hear Chinese perspectives from Dr. George Gao. I'm just checking if is everything set already. Dr. Gao, can you hear us?
Yes, I can hear. I'm ready. Thank you.
Welcome.
Can I share my slides?
Yes.
Okay, I'll share my slides.
Sorry, just a moment. It's opening. It's still a black
Can you see it?
screen. Oh, yeah, but it's not in a presentation mode. It's just...
Can you see my slide now?
We are seeing the overview of your presentation, but not the slides. You should
Still not the slide?
You should put in a presentation mode.
Hold on, let me try. I think I already put it in the presentation, but I don't know why. Hold on a second. Is this okay now?
Not yet. It's
Still not yet.
Yeah, we are seeing very short, very small slides. you should share the window. You should share the window with the presentation. I don't know if you're using two screens.
I don't actually. Let me try again.
Yes, please.
I don't know what's the problem then. Let me try to stop the. Let me try the.
Same thing. No, we cannot see, but
You still cannot see it?
no. What we can do if
I'm
it's,
sorry. Let
yeah.
try. Let me try again. Sorry. Let me stop the sharing and then restart. Let me try restart. Sorry
Okay.
for that. Is that okay now? Still not okay?
It's not. It's still not. We are considering here that when you press share, you have to choose a different screen or a different file, because what we
Okay,
are
let me try again.
what we are seeing is the list of slides, but not the slides properly. So, and okay, now it's better. Yeah. Yeah. Yes, presentation. Yeah, that's great.
Okay.
Thank you.
I'm sorry for that. It's a great pleasure for me to be here. Today, I'm here on behalf of the Chinese Society of Biotechnology. So I was asked to share our experience and lessons we learned for the biotechnology industry in China. As a current president of the Chinese Society of Biotechnology, you know, I'm also the former Director General of China CDC. So I have the capacity really to share with you, for a lot of things. So first, I try to share with you the driving force at biopharmaceuticals. And this time, as you know, we developed our own. I call it China's vaccine. I'm sorry, there's a pronunciation problem here for vaccine. And lastly, I want to share with you my view about the collaboration between China and Brazil. Let me say, for China, as I said, we have a different system, and we always have the government guided by economy and biotechnology strategies. For a long time, actually, under the leadership of the National Development and Reform Commission, and the work is done by my society. We also have this China Bioeconomy Development Report, annual report. So this annual report gave the guideline for the bioindustry development, in China. In my opinion, this is very, very important. This is the stimulus from both the central government and also from the local or provincial government, to guide the bioeconomy or biotechnology development. The society where I am the president of the movement, we all have all these prominent scientists as the leader of this, biotechnology. It was established in 1993. We have the government guided outline. We also have the academic related guideline like the Chinese Society of Biotechnology. So in my opinion, this is very important for our in times of the kind of strategic plan for the whole biotechnology. As you know, the developmental process of bioeconomy industry in China, bioagricultural economy. So no, this is very important. Bioindustrial, so this is another part. At the moment, that because the computer science and information AI driven. So this is also, is guided in China. And more importantly, we are working on the green development. So this green development is very, very important for, for China's whole themes. So when you are talking about the economy, we want to have the green development. So this is, at the moment, in my opinion, is very important in China. Now, our president called for new, we translate this one as a new quality productivity. In my opinion, that's a innovation driven. So China invests a lot of money, funds for the innovation, especially for the society, national institutes, and university. I will give you some examples at the moment. So in China, historically, we address something more about biotechnology. This is more on the academic field. And we, the academic field, will support for the bioindustrial entrepreneurship. And then we are expand more to the bioeconomy. So, this is the general idea about what we are doing. This is reflected in our 14th five-year plan for the bioeconomy. So biotechnology, and for China, we call it the 5-10 trillion pillars of the strategy. First, new generation of information technology, and the historical biotechnology, and green low carbon industry, and high-end equipment, the new materials, and more importantly at the moment, digital creativity. So these are 5 pillars we address more in our 5-year plan annual report. The driving force for the rapid development of the China's biotechnology industry. First, insensitive policy support. This is, I already said, from the central government and also the local government. And then strong market potential. China has 1.4 billion population. So we have that market potential. And also the complete industrial chain. China is such a big country. Though we are part of the global economy or globalization, but China itself, we can really self industrial chain is always there. And fourthly, is a sustainable development by innovation. When you compare with the US, the US biotechnology and manufacturing, they always have their own plan. In March 2023, the White House Office of Science Technology Policy, they released their report, and they are doing similar thing. I call it as 5 specific areas: climate change solutions, food and agriculture innovation, supply chain resilience, and home health, and cross-cutting frontiers promotion. So home health, again, go anywhere. So this is very important. I think, very similar. So with the U.S. system, but the China, the government driven, more important than the U.S. The U.S. the private sectors, especially for the VC, venture capitalists, they are playing more important role than in China. So this is what is the White House, what they have for the low carbon, green, and circular economic development. So they address more for the biomanufacturing. Look at the other areas in the world. For the European Union, they have their European Union sustainable by economy. They are stretching the link between economy, society, and environment. And also, especially for the Germany, they have their own national bioeconomy policy strategy. Again, Japan also, is doing the same thing. Korea, or you heard from the previous speaker, Korea is doing something very similar. Look at what happened in China. I give you a list about what China's national plan. In 2015, 2016, we also have this annual stimulus, for the biotechnology entrepreneurship promotion. So this is very, very important, as I said from very earlier. In the 14th five-year
Recording in
plan,
progress.
and we want to stress the leading role of the national strategy. We want to focus on an integrated supply. We also want to do the manufacturing industry, and we want to accelerate biomedical innovation. We also want to accelerate the integration and stressing the development and utilization of biological resources. You know, biological resources, especially under the Chinese Academy of Sciences, where our Institute of Microbiology is located. And we also enhance biosafety governance. So as you know, after the COVID-19, people always challenge about biosafety and biosecurity. So China at the moment, we address more for the biotechnology field in the capacity of biosafety, and also promote Chinese medicine. And the most Chinese traditional medicine was, raised in a very important role. So this is the structure of China's bioeconomy industry chain. Upstream, this is university and national institutes like the Chinese Academy of Sciences. And in the midstream, we have four major subsectors: biomedicine, biological agriculture, biomass substitution, and biosafety. And the downstream, of course, we have this medicine, agriculture, and sustainable energy. And again, it's the government system. So first, in the biomedicine. In that biomedicine, China has a huge market here. For the last 40 years, this field developed very, very quickly. And the bulk fermentation products, historically, China is very good in fermentation field. Chinese vinegar, Chinese vodka, like alcohol production, all this, it's in a very good form at the moment in China. Again, bio-based materials. So we pay more attention for the bio-based materials. Again, I mentioned about the green development for the bioeconomy, energy. One of my colleagues, they are also working on the biological environmental protection. So this is important. Recently, from my colleague, published in Science, they did from the carbon dioxide directly into starch. This is very important. The paper published last year in Science called Cell-Free Chemoenzymatic Starch Synthesis from Carbon Dioxide. This is really a breakthrough for the theory. So this is from the Tianjin Institute of Bioindustry. So this is very important. In the middle is my colleague, former colleague, Dr. Ma Yehe. This will attract the world leaders and the world scientists to look at this. So this is in general about our bioindustry, including the bioagriculture, and including the biofood, and also for the whole growth in China. We are talking about 30 trillion Chinese yuan by 2027. So the very, very big industry. And this industry, as you know, this give you an overview from 2016 to 2020, and see the growth of this entrepreneurship. So in general, so this is give you some example about this some of the industries entrepreneurship. We give you Henry Pharmaceuticals, Beijing, and everybody, a lot of people knows BGI Genomics, and also Weekend Bio. And just give you some examples for this. This is another example from BGI. They have a sequencers for the manufacturing of this biological apparatus. It's very strong, the whole field. So in the Fiocruz, you already have some MGI sequencers there. Now, I want to come to my second part about the challenges or opportunities under the COVID-19. So, as you know, biotechnology industry and biopharmaceutical industry grows very quickly. Because of that, China also faced a problem for the aging. So this is why this already mentioned about this upstream and midstream. And so this is from the very beginning when we had COVID, and the central government immediately, put their own policy, especially for the China FDA, to push forward for this method for the regulations. And regulations is very, very important. The local government also, they promote their local government for the vaccine development. They have renewed their regulation. So this is a give example. I give you an example for even from my own team. We developed our own protein subunit vaccines. We developed monoclonal antibodies, macromolecules as the antibody, for the treatment. So I called it, George's group. We did from the diagnosis to the vaccine. So this give you an example. So we published the first paper, one of the first papers for the antibody isolation for the homoneutralized antibody, published in Nature. And then we developed this one as a product in collaboration with Junshi and also with Eli Lilly in the US. Actually, a monoclonal antibody isolated from our lab and developed under the US. Though, as you know, we have the US-China conflict at the moment. But once you have some good products or good discovery, they like to collaborate with you. So this is a very good example. And for the vaccine, as you know, we have 7 strategies for the vaccine development for the COVID. First, inactivated vaccine. China, most of the Chinese, we use the inactivated vaccine. It's very popular. Second, live attenuated. This was not developed. Second, protein subunit vaccine. This is in my group, we have the first approved protein subunit vaccines to be used in clinics. Then viral vectored, influenza vectored, and also adeno vectored. DNA vaccine, this time, is not being used, and then mRNA. And in China, we also have the mRNA vaccine, but it's not popularly used yet, and virus-like particle, so it's not developed. Here, you are talking about 4 types: inactivated, protein subunit, viral vectored, mRNA vaccine. At the moment, in China, it's either it's already being used or under development. So this gives you an example. Junshi as a top alliance with Eli Lilly for antibody development. And this is in the book written by Dr. Miller with Özlem Tarici and Dr. Ugur Saying. They developed mRNA vaccine. They mentioned about from my group, we are working on a receptor binding domain, RBD, for the vaccines. So we are using a different strategy. And protein subunit vaccines used 87% with Beck's test population, they use these vaccines. And this is clinical trials and published in New England Journal of Medicine. You are talking about 81.4. Of course, after the escape, the vaccine, we need to develop a new vaccine with a new variant. So this is a new variant, beta and Omicron. At the moment, we keep updating our vaccines. Now, foresee the future, vaccine is very important. What's the next disease? So the WHO mentioned about the disease X. So in my opinion, could be anything in the future. This is the fake public health emergency of international concerns since the 21st century, starting from 2009, pandemic influenza H1N1, until recently, monkeypox. So we are facing so many problems. Of course, Zika, all the Brazilian people might remember 2016, you have a Zika outbreak. So in my opinion, I think from avian influenza virus to Zika virus, you always have this attack from the emerging, re-emerging viruses. And the third part I would call for the China and Brazil collaboration, for the vaccine development, for the emerging virus effect, and also with the BGI, for the development of the any other products. So this is Dr. Carlos Morio, and when he visited China, and we have this statue, Zika virus, and this is 2017. I met your former Brazilian Academy of Sciences President Luis Davidovich, and that's in 2017. I visited Fiocruz with Nicia for the MOU in 2017, and then we had something in the meeting in Shenzhen, that in 2018. Because we have this mutual visit. A lot of my colleagues with the Fiocruz and our vice president of the China Academy of Sciences with Nicia again. And this our interaction for the Infectious Disease Research Center, a joint center between Fiocruz and the Chinese Academy of Sciences and China CDC. For the future, I think academia industry cooperation for China and Brazil, I think our future is very bright. I'm calling for the future collaboration between China and Brazil. I think I will stop here. Thank you very much.
Well, thank you very much for this very informative session where we could see some good experiences. And we have some very 10 minutes time for some discussion and questions. And but first, I would like to highlight what we have here, what we have heard here, which is really important for us, considering where and at what time we are now in Brazil. So we have seen that Korea, with this strategy for self-sufficiency, has a very robust or a very impressive and enthusiastic target of 80% of self-sufficiency in 2030, which is a number that we should take a look and think about Brazil. What is our self-sufficiency regarding vaccine administrations here? We have some quite good examples with flu vaccine, yellow fever, and some good examples here, but we still don't have this number, and it's quite impressive. And also, we have seen the route for Korea on having and establishing a hub for biotechnology, right? Having establishing a training hub where they export knowledge to other countries, which was really important strategy, and also developing CMOs in Korea, right? Which is something that in Brazil we have just started, right? Recently we have the launch of the first public CMO in São Paulo State, and also we have the private CMO also that was launched this year. So which is something that we have to think. We have, for example, in Butantan, we have about 100 researchers for new biotechnology products, which are in early stages, where we need places to build the bridge to products that could be used for clinical trials and for experimental steps for regulatory approval, where we don't have all the capacity to do that without using CMOs. So this is something that we have to consider. And also in China, very impressive numbers and very impressive strategy, 5-year strategy for this, what was called, which is very interesting, bioeconomy. Is not only focusing on a small part of the biotechnological product, but also China has the impressive possibility to control the whole chain. We know that when we think about when we say we have self-sufficiency for flu vaccine or for yellow fever vaccine or other vaccines we do here, we know that we still depend on a lot of products or equipments or reagents or a lot of things that come from external countries, which in many cases is China or European or US. So how independent we are if we don't control the whole chain like China is aiming. And in the pandemic situation, we have seen that, right? Lacking a lot of basic materials for the self-sufficiency. So this is something you have to consider. Doing that, I would like to ask one question for you, Chris, which is considering what you have studied from Korea and how you have shown that investments come from government and also private. So how do you see and which kind of advice you would give on balancing these funds that come from government or private, right? Brazil, we have a strong now politics on stimulating from government the self-sufficiency, but what percentage this should represent in a good strategy for the self-sufficiency and for an example like Korea?
Well, that's a really tough question. I don't think I can talk about percentages. I think going back to Korea, what you see, I would say the government funding goes a lot into manufacturing. So as they built the first CMO, then it was strengthening clinical trials. Now again, vaccine manufacturing on the large scale. The private investments from the companies, there were times when 50% of the wins would go into R&D in the company. So a lot of the innovation in the companies were paid by the companies, and I would say the government funds went more towards the whole strategy. Where do we want to go? What facilities do we need for that? Relying a little bit on then also academia, the small companies, people having bright ideas to that the innovation comes, let's say, independently. But then there's also the right fund. You cannot forget, which is in between the public and the government, so there's another source of funding to support the innovation, I would say. But about percentages, I cannot speculate.
That's right. No, interesting that you say that these funds, which in Brazil we have very limited applications. We have very few applications. When we consider Right Fund or Bill and Melinda Gates Foundation, which is represented here, and other funds that could help us not to depend only on the governmental money to jump this. An important highlight that you mentioned also was the prioritization. Korea decided and selected vaccines, some vaccines that they aiming to do and make self-sufficient. It's not all of them. It's some of them which is important for that population, right? This prioritization is fundamental. And the team here that used to work with me in Anvisa knows that we always used to say, when everything is priority, nothing is priority. So we have to keep this in mind, otherwise we'll reach nowhere. And Dr. Gao, just to finish, and I want to hear and also ask for a word of advice to our country, considering what you have seen in this bioeconomy policy in China. So what would be your advice to us, Brazil, to aim for self-sufficiency and to control the chain, the whole chain, as you mentioned, as a pillar for bioeconomy?
As I said from the very beginning, China have our own system. So this is why we have the whole chain, as a closed circle in China, because we have a different system and plus a big population. So in my opinion, Brazil, is your system is different, from China. My advice for the whole by industry or by economy. Maybe Brazilian government would try to figure out and find out, the gaps, and what you would put your chain as a closed circle to which one you should, under such a globalized world, which part the Brazilian government should invest more or encourage the VC to invest, which one you should collaborate with some country like the US, China, or any other European countries. I think this needs some kind of strategic plan. So I think this is very important. Thank you.
Thank you very much. Well, I have seen the 2 minutes sign, so I believe we could take one question from the audience, if there is anyone who wants to ask. I'm not seeing any raised hands. Renato Strike, please.
Thank you. Renato Astray, Instituto Butantan researcher. So I would like to highlight one common feature of this, the two experiences and one difference. The common is that we saw two very strong government investments in both of the cases. And one difference is that in China we have a big market, but not in South Korea. So I think that in South Korea example, they had to focus not only in their own market, but in the outside market also. So my question is, in both cases, maybe, how the government could make the policy to the different companies don't overlap their portfolios of vaccines and develop this. What was the policy? Because we see that in Korea, we have this kind of sequential programs that were being done, but I think that if the pharmas were competing with the same product, it would be very tough to reach.
Well, who wants to take first? Chris?
I'm not sure. I have to admit I know the answer. I mean, the Korean biotech is really coming up now. It's not like a landscape that you would have a GSK, Merck, and Novartis in the same country competing. So, but to be honest, I don't know how they decided maybe who will work on the HPV, who will work on the other one. So apologies for that.
Dr. Gao?
Yes, and for China, as you know, this time, give you an example for the COVID-19. You have almost 100 small business. They entered into this COVID-19 vaccines. So many US VC, they got into this, I call it very, very narrow tunnel for the vaccine development. So from that, the government would encourage for that. And meanwhile, in my opinion, that's lessons we should learn. And so after, after these 4 years, at the moment, we have some VC, they need to go back to see what the resources they have already, wasted. So government encourage more VC to get into some small business. Meanwhile, you might have some small business, they waste their resources. So this is something in China, we need to have a more or highly regulated policy to see the future. Thank you.
Well, it's clear that in the case of Brazil, where we have limited resources, we have to consider this. This is an important question because overlapping is competing resources which are already limited. So we have to know and we have to establish a policy. It is important here to know where we can go, where we should go, and how we avoid this situation. Well, with that, I'd like to thank Chris, thank Dr. Gao, thank you for the audience, and see you in the next session. Thank you.
Thank you.
Obrigada, Gustavo. Parabéns aos participantes.
Thank you, Gustavo. Congratulations to the participants. I would like to invite now for a poster presentation, Nájila Santos Pacheco, de Campos. Please, Nájila.
Ela vai apresentar o pôster.
She's going to present the poster Anti-CAAX CAR T cells in the treatment of renal cell carcinoma patient-derived xenografts.
Eu pediria só a compreensão
I
desse...
would ask your understanding, you who are leaving the auditorium, we have a poster presentation, and as I mentioned yesterday, it's very important. They are prepared very much for this presentation. I would like you to be silent, please. Thank you very much.
Olá, boa tarde. Eu sou a
Hello,
Nájila.
good afternoon. I'm Nájila. I'm a doctorate student in the program, graduation program at the University of São Paulo, and I was oriented by Professor Eduardo Soares, and this my paper is anti-CAR T cells in treatment of renal cell carcinoma patient-derived xenografts. So I'm going to talk initially regarding renal cell carcinoma. It is the 7th most common carcinoma in men, and it has a subtype that is also common, which is the carcinoma of clear cells, and it represents 75% of all carcinomas, and it has a very bad prognosis, metastasis, and the average of survival is about 13 months. And from there, you have the necessity to develop new therapies for the treatment of this carcinoma. As I mentioned, we produce T cells. It's a mechanism of a membrane, and it's overexpressed in several hypoxis tumors, solid ones, and it's a constitutive of this clear cell carcinoma. So this is the target of our CAR T cells. So a bit of this CAR T cell therapy is one as we have T lymphocyte expresses an artificial substance, and it's associated with an antigen with tumoral cells, and it has some domains with CD28 of transmembrane and extracellular, which is associated to the antigens present in tumor cells, and also the intracellular domain that has two parts, a very important one that has the CD28 and also CD30. And this therapy has been developed and with very great results, but we have challenges for the solid tumors because these cells are exhausted, and there also has issues regarding migration of the solid tumors. So thinking about the challenges that we could find, we worked with a model that is a patient-derived xenograft, and this model presents a great mimetization of the tumoral environment where we can check in real time new drugs for the patients. Well, the objective of our work is to verify the efficiency of the efficacy of this kind of T cells with co-stimulatories of C21 domains for treatment with this model of PDX and also using low doses of CAR T cells. And these experiments are long when they have the duration of 6 months, and we start with implantation of 3 to 4 fragments of tumors from patients, and we did a weekly follow-up through using ultrasound, and we injected CAR T cells with the animals. We did 2 infusions and also verified the groups for treatments that for control, there's T lymphocytes that do not have artificial receptors, and other 3 groups of therapy, 2 with the certain specificity that you can check on the slide, the details, and also with the CD28 for the cells, and you can verify the growth of the tumor, and we also saw impacts in the life cycle and survival. So the first issues that is relevant for this kind of trial is the characterization of the CAR T cells, and we can see on graph A, the proliferation of these cells were excellent. They grew very much, and the levels of transition went over 90% in all the treatment groups. And after 14 days, these cells are still expressing the artificial receptors stably in the average of 40%. And after 14 days from the infusion, we took the blood of the circulating blood from these animals, and we verified that these cells were there, transducted and proliferating. So we here present the tumoral growth from the variation of the area with the ultrasound follow-up, and we see use 2 PDX patients and also CAR T cells, and in the CD4, CD8 cells, they presented a robust growth. The control cells grew immensely, and this could refrain the tumoral growth from and it's important that this construction is better for this model comparing to the control group. So we verified the survival curve, and in the panel on the left, and the treatment with the columns and the treatment. And in this case, we verified that the CD28 group, we did not find any metastasis with the animals that we used for this group. And for the second group, they had smaller tumors, a reduction in size, and they also have better survival rates.
Algumas moléculas para marcadores de exaustão dos linfócitos, né? Foram LAG3, CTLA4 e PDL1, e verificamos que a construção CD28, 2:1, células CD4...
We ascertained that the CD4 and CD8 featured a robust reduction in these 3 markers, so that they almost blocked their presence was almost blocked. Then we've also ascertained toxicity. We know that the therapy has a question of being very effective, but as a counterpoint, there's a problem of toxicity in the patient's body. So we checked the creatinine and transaminases to ascertain toxicity in the liver and the kidney. And we saw that they, in our treatment groups, when compared to the control group, didn't have many alterations. We concluded that there was no noticeable toxicity in our treatments compared to the control group. Thinking of from the prospect of our work, we were able to reduce the tumor growth, basically in the 4-1BB, the CD4, CD8. In addition, this construction didn't indicate any toxicity. for animals. And in the super challenging model, which is the PDX, very troublesome, we hope to continue this work from the perspective of increasing the dose to look at the effects on tumor growth and the metastasis of these animals. In these animals, we also hope to select patients that express carbonic nine-hydrases and PDL1 to assess from other constructs, a research group that works with these therapeutic groups, but these are KT cells that can secrete antibodies to ascertain mostly the alterations and immunological checkpoints. Well, I'd like to thank my mentor and all my colleagues that took part in this very challenging work, especially because of the PDX model. And thanks for the financial support from FAPESP and your attention. Thank you.
Obrigada, Nájila.
Thank you, Nájila. We would like to invite on stage Leonardo Ribeiro Batista Silva for his poster presentation. He will present the poster, Efficient Large-Scale Point of Production of Affordable and Anti-CD19 KT cells for immunotherapy. Good afternoon, everyone. First of all, I'd like to thank the organizing committee for the invitation and for giving us the opportunity to prepare the work we've developed at the National Cancer Institute. And today I'll be talking to you about one of the projects that we have developed over at the National Cancer Institute, which is the production or manufacturing of KT cells at the site where patients will be eventually treated. So Nájila has already helped me with some concepts, but now going back briefly, what is the CAR? The CAR acronym, antigen chimeric receptor. And that's so because it's an artificial receptor that is a chimera in which you have qualities or characteristics of the T cell receptor with the antibodies. So you have a part of, SC part, FV part, which is able to recognize the antigen, tumor antigen, and a domain, intracellular domain for the activation of these T cells. And how does T cells get produced? Basically, you get the cells from a patient, you modify them genetically so that they'll be able to express the chimeric receptor. You expand these cells in ex vivo fashion, and then you reintroduce those cells in the patient's body. Currently, there are 4 products that are commercially certified in Brazil. These are products based on CAR KT cells, anti-CD19, and BCMA, which is CAR. Well, since we're at a symposium held by Bio-Manguinhos, I'd just like to highlight that we have a technology transfer agreement with Carencross, and these KT cells will be produced at Bio-Manguinhos through a partnership agreement with the National Cancer Institute. Now, drawing on this, on the viral vector production platform for genetic modification of the KT cells, they'll be produced at Bio-Manguinhos, and we'll be able to do this manufacturing also here in Rio de Janeiro. Well, today, the process of manufacturing has these features. It is based all the way from the leukapheresis of the patient, the preservation of leukocytes, and then you send these cells, frozen cells, to the site, to the manufacturing center. These cells at the manufacturing center, they are activated, and you expand these cells and you modify them genetically using viral vectors. this thing is working by itself on its own, I think. So upon genetically modifying these cells, you expand them ex vivo, and these cells go through a quality control process, and they are taken back to the hospital where the patients will be treated, and the cells will be infused in the patient's body. That's how the manufacture of KT cells takes place in Brazil. This is how it happens. Now, the goal of our work is to be able to align and to do the manufacturing of these KT cells efficiently and large scale at the site where these patients will eventually be treated, which is the National Cancer Institute. Well, our strategy to do that is not to use viral vectors. We're using non-viral strategies, transposon, transphorases. We have two components in the system. We work currently on two components, plasmidium components, one of which codifies transposon, and the other one is transposase, and the other through a mechanism of copy and paste in the genome. We're able to insert these transgenic cells in the genome of the KT cells. So there is an advantage and a disadvantage. There are pros and cons when it comes to the use of viral vectors. For example, I'm pointing here the disadvantages and the utilization of viral vectors. You have a production which is very cumbersome, very troublesome, and high cost. And the whole plant, the production costs are high. The whole plant that was set up to produce vaccines at Bio-Manguinhos based on viral vectors, it will be turned into a venue for the production of vectors for the transduction of KT cells. And we have some risks also of activation of oncogenesis when you do this, especially when it comes to viral components. And commercial products use this kind of system of viral vectors. So we began, first of all, to do some small-scale data in which we showed that we're able to generate these cells, and these cells are efficient in the control of the tumor. And we're showing here that these cells, 19 BBZ, are the cells with KT. So we have here the animals have been inoculated with a tumor that's able to, a tumor cell that's capable of expressing luciferase. So we have the bioluminescence here following up on the tumor progression, and the animals that were treated with KT cells. You have here the regression and the progression of that tumor. When we look at the survival rate of these animals, we see that the animals treated with KT cells generated, even in small scale, have the ability, anti-tumor ability, effective anti-tumor ability capable of increasing the lives of these treated animals, even after a long time. At the time of euthanasia, these animals, the ones we've inoculated with KT cells, they're still present in the bodies of these animals. So the idea of this project, obviously, is to make sure that our genetic modification tool and the manufacture of KT cells, to make it viable, to make it feasible, and now increase the scale of production of those cells. So the flow, the production flow for those cells is through peripheral blood from patients. We do electrophoresis of those blood cells, and we use those bottles, 1 liter bottles called Rexim bottles, in which we're able to produce a large amount of cells. So the first outcomes, large-scale outcomes, have shown us that we have an important efficiency rate here at transmission of those cells, in which over 50% of those cells in this experiment were positive to express the chimeric receptor. Sorry, this is working on its own. It's not me. It's the gizmo I'm holding. So for us, we were able to achieve an important number of T cells at the end of the process of expansion. And these T cells have a total number of T cells carrying our chimeric receptor. And then by using the PDX model, we're also able to inoculate those animals with a primary tumor derived from patients that had leukemia for example, acute lymphoblastic leukemia at the National Cancer Institute. And these immunodeficient animals were able to develop the tumor. And upon treating those animals with the CAR T cells, we were able to see through the flow scintigraphy rate, we have the positive CD cells, and there was a reduction in the frequency of those cells in those animals. And finally, we measured the survival rate of those animals treated with CAR T cells. In this model, cells derived from tumors in patients, and there's been a significant survival rate, significant survival of these animals that were treated with the CAR T cells generated according to the large-scale protocol. As a prospect for our work, we were able to generate these CAR T cells with an important capacity, anti-tumor ability, in an amount of cells that is sufficient so that we can treat patients at the site where these patients will be catered to, will be cared for, which is a cell processing unit located at the National Cancer Institute of Brazil at low costs compared to the CAR T cells generated through viral vectors. So I'd like to thank our group members for the assistance and the foment organizations. Thank you so much to financing agencies. Thank you so much to all of them. Thank you, Leonardo. Congratulations on your presentation. Well, now moving on with our presentations, I'd like to invite as the chair of the next panel, Advanced Therapy and Public Health and Unified System Technology and Access, the coordinator of the National Institute of Science and Technology and Medicine, Regenerative Medicine, and the National Network of Cellular Therapy, Antonio Carlos Campos de Carvalho. Is a physician from the Federal University of Rio de Janeiro. He has a PhD and a master's in biological sciences and biophysics from the same institution. He has had ample experience in physiology and biophysics with emphasis on cardiovascular physiology, acting mainly on the following topics: communication, intracellular communication of stem cells, and cellular therapies. He was a visiting professor at the Einstein College in New York. He is an emeritus professor at UFRJ, a member of the Brazilian Academy of Sciences and the Academy of Sciences in the Developing World. As the debater, I'll invite the Vice President of Production and Innovation in Health at Fiocruz, Marcus Aurelio Krieger, who will be coming to talk to us virtually. He'll be speaking to us on video. Has a PhD in biophysics from the UFRJ, has a BS from Federal University of Paraná in biology. He has been working in molecular parasitology and biology, molecular biology, working on the trypanosoma cruzi, genetic expression, functional genomics, cellular differentiation, and the use of molecular biology technicians towards the development of tests and diagnoses. It was the adjunct director of development, technological development, prototyping, and production at the Carlos Chagas Institute at Fiocruz. Is a technical coordinator of the production unit at Fiocruz for diagnoses of nucleic acid. As speakers, we would like to invite Professor from Paramount School of Medicine from Pennsylvania University, James Wilson, who will present to us through a video EASI-based vectors as a platform for treating disease. His career has been devoted to rare diseases and ways to treat them through genetic therapy. He's a medical professor and pediatrics and a director of genetics at the University of Pennsylvania. He led efforts to develop the field of genetic therapy. Most recently, he focused on vectors, improving gene therapy and clinical applications, and the addition of genomes mRNA therapies as well. I would like to invite on stage the co-founder and CEO of the Karen Cross, Mauro Droccoli. Please, sir. You'll be making the initial presentation, lentivirus vector-based solutions to treat human diseases.
University of Western
His PhD from the University of Australia and MBA from Johns Hopkins University. He has had experience in the design and development and the manufacturing and technical translation and regulation, implementation, clinical implementation and commercialization of vector technologies and lens viruses. He founded the company that developed the first genetic therapy product approved by the FDA and co-founded Caring Cross. And researcher from the National Institute of Cancer Institute, Martin Bonamino. Gene therapy-based approach to treat patients at the public health system. That's his presentation. PhD in biological chemistry from Urford J and BS in biological sciences at the same institution. He has had experience developing genetic therapy protocols and immunotherapy against tumors, in addition to acting on the immunological characterization of cancer. He is a Fiocruz expert, where he is one of the coordinators of the Fio-Cancer Network. Antonio Carlos, please, you have the floor. Good afternoon, everyone. I will switch to English because the panel is international.
And to be able to introduce leading researchers in the field of advanced therapy products. As all of you know, we're seeing a true revolution in medical care, especially for diseases for which there were no previous effective therapies. And in this panel, we will have two world leaders in the field of gene therapies. One that deals essentially with in vivo gene therapies, and the other one that has developed essentially ex vivo gene therapies. Additionally, we'll have a leader in Brazil in the gene therapy field. As you've seen in the previous presentation by his laboratory, Martin Bonamino is one of the few researchers in Brazil developing gene therapies for the public health system. So not only are they leaders in the field, but they're trying to resolve I think the main problem that the field of advanced therapies have right now, which is the price of those therapies. And that's the main reason why Fiocruz has signed already a technology transfer contract with Caring Cross, headed by Professor Dr. Boro Doprolich, and is about to sign such a contract with Dr. Jim Wilson from the University of Pennsylvania. So without further delay, I turn to, I think the first presentation will be by Dr. James Wilson from the University of Pennsylvania.
Hello, my name is Jim Wilson, and it's a pleasure to be invited to the 8th International Symposium on Immunobiologicals from Fiocruz. And today I've been asked to talk about AAV gene therapy for orphan diseases. A brief background on AAV vectors. There are 3 generations of AAV vectors. AAV first generation were derived from isolates of AAV discovered in the 1960s from laboratory preparations of adenoviruses. At the turn of the century, my laboratory substantially expanded the repertoire of AAV vectors through the identification of AAV sequences from endogenous primate sources. These vectors have names such as AAV8 and AAV9 and form the basis of the majority of vectors that are currently under clinical development. More recently, there have been efforts directed to the development of AAV vectors based on engineered versions of the AAV 2.0 vectors. Today, I'm going to share with you the data that we've generated in clinical development of AAV 2.0. The talk today focuses on the use of gene therapy to treat CNS disorders. The challenge is delivery. The holy grail of gene therapy for neurologic disorders is injecting a vector into the blood to target the CNS crossing the blood-brain barrier. That has been a very difficult challenge, so the early aspect of the field evaluated ways to inject vector directly into the brain through multiple injection sites. We've instead focused on broader distribution of AAV by injecting the vector into the cerebral spinal fluid, either through a lumbar puncture, into the ventricle, or into the cisterna magna, and it is the cisterna magna approach that we have used in all of our clinical development programs. When we think about gene therapy, I suggest we should think about it as a platform in which we leverage the power of the delivery and the manufacturing platform across multiple diseases to improve the probability of success, simplify clinical development, decreasing the cost, and improving timelines. Our CNS platform uses a clade F vector Hu68, very similar to AAV9. The vector is manufactured using an adherent 293-based transfection platform in a iCELLis bioreactor and injected into the cisterna magna to get broad distribution throughout the brain and the spinal cord. The therapeutic applications that we are pursuing are in the treatment of global neuropathic diseases, in which gene correction in a minority of cells of the brain can have an impact on adjacent cells in the so-called bystander effect. We have a number of lysosomal storage diseases in clinical development approaching commercial. A company we started is using this approach for acquired adult dementia that is monogenic. We also have programs ready for the clinic in neurodevelopmental diseases and CNS malignancies. This approach is also useful to target cells of the spinal cord, the dorsal root ganglia, and the motor neurons. And we're developing this for next generation SMA1 and 2/3, Kennedy's disease, and ALS. Our most advanced aspects of the CNS platform are described in this table, and they are diseases that affect storage in the brain, so-called lysosomal storage diseases. All of these are clinical stage. The most advanced is MPS II, that has finished pivotal studies in collaboration with the company REGENXBIO. And we also have phase 1/2 data for MPS I and GM1 that are entering pivotal through REGENXBIO and Passage Bio. And then Krabbe and MLD that have open INDs and are enrolling patients now. I'd like to share with you data from a few of these examples. The first is GM1 gangliosidosis. This is due to a defect in the enzyme beta-galactosidase that leads to accumulation of GM1 as the substrate. This disease has various phenotypes. The most common and most severe are early infantile, diagnosed less than 6 months of age, late infantile, and juvenile. The storage pathology as a result of the accumulation of substrate leads to this kind of picture in the brain that is associated with rapid neurodegeneration. The natural history of infantile GM1 is that developmental milestones are not achieved. There is some somatic manifestations, hepatomegaly and coarse facial features, but as a result of the neurodegeneration, hypotonia, and early death. This is a meta-analysis of many, many studies evaluating the natural history of infantile GM1. That is a diagnosis less than 12 months of age. And what we have here is onset of the disease early. These are months, but you can see almost complete mortality with 50% mortality somewhere around 20 to 24 months, 2 years of age. So very significant disease with significant unmet need. We're developing a gene therapy using our platform to show that we can diminish the accumulation of substrate and lead to increased survival. So at the University of Pennsylvania, we conducted a natural history study, a prospective study in early infantile, late infantile, and juvenile, where we evaluated GM1 neurofilament light chain in the CSF and plasma neurofilament light chain. And you can see that the substrate is very highly accumulated in early infantile, less so in late infantile, juvenile. Also a marker of neuro damage, elevated in early infantile, reduced in late infantile and juvenile. And when we look at the survival of early infantile with these accumulated biomarkers, you get complete death by 40 months of age. Late infantile is associated with less mortality, and juvenile even less. So a direct correlation between GM1, neurofilament light chain, and survival, suggesting these would be good biomarkers for an accelerated approval pathway. So I just want to share interim phase 1 data in our clinical trial that we're conducting with Passage Bio, where we have dosed 4 subjects at low dose, 4 at high dose, and now actually this is a medium dose, and then 3 at a higher dose. And measuring the CSF GM1, which is not reduced in low dose, but consistently reduced at the next higher dose, approaching normal levels. When we look at all patients enrolled as with a diagnosis of infantile, less than 12 months of age onset, and compare their survival to natural history, we get complete survival, showing a dramatic clinical impact, and really excited about moving this forward very rapidly into pivotal and then commercial for global access. Another leukodystrophy that we have open studies with, enrolled a subject, is Krabbe, early infantile Krabbe, severe, significant neurodegenerative disease, diagnosis less than 6 months of age, and prognosis death 2 to 5 years of age. This is the enzyme that's deficient in Krabbe, and it leads to an accumulation of substrate in particular cyclosine. As I mentioned, we've dosed the subject so far in enrolling, hope to enroll others soon that showed good biomarker correction, but I'd like to share with you the compelling preclinical data that we generated in support of this clinical trial, and that is a dog model of Krabbe disease that has the same phenotype, early mortality within 2 to 3 months of age, and demyelination, as you can see by this Luxol blue stain in the brain of the Krabbe dog, but not the wild type, with the absence of staining. Clinically, we've evaluated untreated dogs with Krabbe, normal dogs, and then treated dogs. So in the untreated dogs, that they have reduced nerve conduction velocity, but they need to be euthanized before this progresses. Nerve conduction velocity is normal in the treated and then normal animals. And the untreated animals have an accumulation of CSF cyclosine, a great biomarker, whereas this is not accumulated in normal in the AAV treated animals. So what about the phenotypic correction? These are 2 dogs from a litter, one that has Krabbe, as shown in this movie. At 12 months of age, sham treated, you can see the animal's tremulous and trying to move, and lost motor function in its hind legs and had to be euthanized very soon after we evaluated the animal. A sibling from this litter was treated soon after birth through an ICM injection of vector. And this is the animal at 82 weeks. You can see no hint at all of motor dysfunction and essentially a normal, vibrant, happy dog. So it's this kind of stable, efficient correction that supported us moving into the clinic. So I want to talk about a really exciting product that we're developing, and that is an improvement on the drug called Zolgensma, which is an AAV9-based gene therapy to treat SMA type 1. SMA type 1, again, is a very severe early infantile onset disease. It's not a storage disease. It's a disease of a loss of function that affects the motor neurons. Diagnosis before 6 months of age, 75% mortality by 13 months of age, will not achieve milestones. A product was used called AV9, based on a vector discovered in my lab, AV9, correcting the genetic defect in motor neurons by infusing high doses of AV9 into the blood, very high doses. That does lead to a substantial improvement in the prognosis of these patients. So that the dose is very high, 1E14 per kilo. It does help the patients, although associated with really substantial systemic toxicity. And the cost of vector is almost $250,000 per dose, and it's too high to treat the attenuated version called SMA 2 and 3. This is approved in many geographies. The asking price is over $2 million, and some health authorities can negotiate a lower price, but incredibly effective, associated with high cost and a lot of toxicity. So we're in the process of developing a next-gen AV gene therapy for SMA that is improved in a variety of ways. We have an improved vector, and we are proposing to use an ICM delivery versus an intravenous for SMA1. These are studies done in a non-human primate, injecting a vector intravenously, and then ICM, looking at the central nervous system. ICM delivery with a 10-fold less vector is 50-fold better, and as you would expect, almost 1,000-fold less targeting the liver, indicating a likely better safety profile. We've also optimized the vector so that we've improved the safety of the promoter and put in a sequence that can circumvent another toxicity that's been associated with first-generation products, which is sensory neuron toxicity of the dorsal root ganglia. And we show that our improved vector here shows great survival in comparison to other vectors in an SMA model of mice. So this is an improved efficacy and safety in SMA1. The cost of manufacturing can be reduced 10-fold, has the potential to be useful for SMA 2 and 3. We can leverage this experience in other sensory motor neuropathies, SBMA, which is Kennedy's disease, Charcot-Marie-Tooth, and ALS. So to conclude, we're really excited about working with Fiocruz in Brazil and to identify ways to get these products approved and distributed and access to patients. And the way that we hope to do this is to create worldwide centers of excellence for our research and commercial enterprise, where the treatment is performed once at qualified centers, and we partner with governments, key opinion leaders, patient advocacy groups, social media, to try to simplify the sales and marketing and the commercial outlay. And included in our early network are regional centers that we will leverage for a broader access and development with Philadelphia, United States, Brazil, working with Fiocruz and the Minister of Health, also London, Saudi Arabia, and South Korea. So again, I want to thank you for inviting me to participate in this incredibly exciting symposium, and looking forward to our growing collaboration with key stakeholders in Brazil to bring curative gene therapy to patients suffering from rare diseases that are resident to Brazil. So thank you very much.
Now, thank you, Jim, for an excellent presentation. And now we have Dr. Boro Dobrulich presenting as what Caring Cross is doing, and especially what it can do also in collaboration with Fiocruz in Brazil.
Boa tarde. It's a pleasure to be here, and I thank Antonio and the organizers for inviting us, and not only to present here today, but we're so excited at Caring Cross to be able to collaborate with Fiocruz and Bio-Manguinhos on our collaboration of tech transfer, which I'll tell you about in the presentation. So for those that you don't know, Caring Cross is a non-profit organization, but we really run mostly like a biotech company, in the sense that we develop technologies, implement them, and form collaborations to distribute these technologies to our partners around the world. Okay. Yeah. So this is just to give us a grounding how clinically successful many of these products are. These are the ex vivo cellular gene therapy products, and on the left there you'll see about 6 approvals in the CAR T-cell therapy that have been transformative for many patients that have received them in various oncology indications like leukemia, lymphoma, multiple myeloma. And on the right there you'll see the stem cell gene therapies, in which there are treatments for genetic diseases, but also for sickle cell and beta thalassemia. We are focused on these autologous therapies because they really work very, very well. What these autologous therapies are, you're taking cells from a patient, you're then genetically modifying them, now usually almost exclusively with a lentiviral vector, and then those cells are then reintroduced back into the patient. And we focused on autologous and single donor therapies. You can have a donor, but in this kind of very fast manufacturing process, because these therapies really work well. There are other therapies on the horizon, like allogeneic and IPS, but they are very far into the future in terms of how they compare in terms of their safety and efficacy compared to autologous. So we're really focused on autologous and making this paradigm shorter, simple, more economical for delivery to the broadest number of patients that require them. But their current price is very high, right? Kymriah is $475,000, Yescarta $375,000, and Zynteglo, a treatment for beta thalassemia, is $2.8 million. This is an extraordinary high price, and it includes only the product cost, and it's really not sustainable over the long term, even like countries like the US and Europe. Right now, there's a very limited number of patients that are being dosed, but when these therapies become mainstream, tens of thousands of patients, there's not enough money in the system to really support this level of pricing. And it's certainly not feasible in low middle income countries, right, obviously, and it really restricts access, particularly for underserved populations. And those underserved populations are not just in low middle income countries, they're also in the US. And this is just a data point here that was published, where we're looking at the CAR T-cell therapy utilization by race in the United States from 2016 and 2021. And what we find is that about 80% of the patients receiving CAR therapy were white, where about the median population in the US that is white is 60%. Clearly disproportionate access against those patients who are underrepresented. And the global distribution beyond the developed nations, the high-income nations, let's say, there's no significant access, as we know, because of the high price. So why is there such a high price? Well, It's a complex logistics. When pharmaceutical companies own these products, they want their own logistical network and systems and their own factories to produce it, and that's quite understandable. And so you're shipping the product. Let me see if I can get this to work. Yes. So this means you're shipping the product from the hospital first to the factory, and then back to the hospital. And usually there's one facility per continent, right? So it's very long logistics. And this complex logistics and the payment logistics that go on between the hospital and the company and the insurers is, with the central manufacturing facility, is very expensive to establish. And because you need such a high number of personnel to maintain these facilities, it's very expensive to maintain, particularly the chain of custody, chain of identity. And because each company wants to make its own infrastructure, its own manufacturing facility, that manufacturing facility has a high capital cost, and that capital cost needs to be put back into the final product cost, and that's why they're very expensive. And the patient result is not all that exceptional, let's say. When you have to cross state and country lines for the approval of these payments, that significantly delays access to patients. Also, vein-to-vein times, it's been reported that in the US, it's 56 to 85 days average. And what that means is that many patients with advanced disease don't get these therapies in time. They die while the therapy is being made. And that's really, I think, unacceptable. And also, it's difficult for patients to travel long distances because of their families to large treatment centers to get access to these products. So right now, it's really not optimal for the patient. So how do we think about reducing cost and enabling affordable access? Well, we think about it in terms of, on the left there, you can see point-of-care manufacturing, where the product is made at the clinical center or very close to the clinical center. Point-of-care manufacturing is significantly much more economical than centralized manufacturing. In fact, it can reduce the cost by a whole magnitude, right? And it certainly lowers the carbon footprint. Also, there are no cross-border payer issues, which means the patients can get the therapy fast. You don't have to go approval between an inter-country payment or an interstate payment, for example, in the US. And the vein-to-vein times are generally very short, about 7 days average, but we can see also how they can be 1 day in the future. And it's better for hospitals because when the hospital is making the product locally, or has access to the product locally, it can schedule the patients better. So those patients that come in with more advanced disease, they can be prioritized compared to those patients with less advanced disease. So it's much better for patient scheduling when you make the product locally. And many will tell you that hospitals can't manufacture these products. It's always companies that manufacture the products. And that's actually not true. Actually, hospitals have had BLA-approved products in the US. In fact, they make cord blood under FDA approval. And they're certainly capable of manufacturing CAR T cells. Many of the centers that pioneered this field manufacture these CAR T cells at their facilities. So it's not just about point of care, but it's also about improving the technology, right? We're focused on short and simple manufacturing processes, in contrast to where many companies are going to very large, sophisticated, automated systems, which actually doesn't help in terms of the cost. The cost's still high. You maybe save a little bit on labor, but a lot of the material cost becomes very, very expensive. So what we're focused on, as you'll see in the presentation, short processes, simple processes to lower the cost, and integrating automation when necessary, right? In some cases, it's really not necessary to produce highly functional, safe products. If it's necessary in the future, we certainly can implement them. So the first thing that, when you think about point of care, that the regulatory authorities will ask is that, can you manufacture products, consistently throughout a network? And the FDA and other regulatory authorities have been looking at this very closely, and they've been very supportive of it. On the left there, you'll see that, in fact, a dossier from the FDA on point of care or distributed manufacturing for drugs, seeing that one site is a very weak supply chain. Having multiple sites is very, very useful for redundancy in the supply chain. And in the middle on the top there, you'll see the sweet spot for point of care manufacturing, and that's autologous cells. When you're taking cells from a patient and putting them back in, well, it's a sweet spot for point of care manufacturing because you want a rapid turnaround time. And there's some other data. If you look on the bottom right there, I'm not going to go into it in great detail, but in Spain, they've already developed a network They've developed a CD19 car that's produced in multiple hospitals around the country and is being insured, paid back. The product is successfully being made. So certainly this is being looked at in other countries and is being successfully implemented. We published a couple of years ago, seeing the need to have data that you can show comparability between different sites. We had a clinical trial with a CD19 car with two very disparate sites, one in Cleveland, Ohio, and the other at that time in Moscow, Russia. We couldn't do this study, obviously, today. But we had a generic car 19 that we created, and then in both sites, we had the same instrument, the same materials, the same media, the same release reagents, and we found that in both sites, the products were highly comparable, even though the Moscow site at the pediatric center was treating pediatric patients and the Cleveland site at Case Western was treating adult lymphoma. So we showed in that paper that the product is very, very similar, and the vein-to-vein time was 8 days, which meant that every single patient that was enrolled got dosed, got treated, and they all had good outcomes, showing again the benefit of point-of-care manufacturing and making things rapidly and locally. So this is a little bit more color around those clinical trials. You can see on the top left there, that's the team at Case Western. And at that clinical trial on the top right, you can see that we had an overall response rate of 82%, which is great. At the pediatric trial on the top right there, there's two different patients from two different families enrolled on the pediatric trial. And at the pediatric center in Moscow, a 90% overall response rate. Great. And I'll focus your attention just briefly on the bottom panel. They were very excited. Obviously, they had a press release where we did a bispecific CAR and targeting two antigens on the leukemia and lymphoma cells. Because what we know is about half of the patients after receiving CAR T-cell therapy relapse. They lose the antigen, right? So when you have two targets, it's much more unlikely for the tumor cells to escape. And in that trial, we had a 92% complete remission rate and only one patient relapse after 2 years, right? So we were very excited by that. And we're going to continue that theme. You're going to see that the Caring Cross products that we are developing is a triple CAR, a 19-20-22 CAR to really address the issue of relapse long term, to put it to bed. So when you think about a point of care manufacturing, you think about facilities, right? And the facilities that you can have on the top left up there are either clean rooms that are already existing in hospitals, right? Hospitals have them, they can use them to manufacture CAR T. But there are hospitals that don't have them, right? They don't have access to clean rooms. And so we're working with a company in Florida that makes these mobile clean room units on the left in the middle there. Let me see if I can get this. Yeah, there, right? Germ-free makes these mobile units, which you can actually put them in the parking lot of a hospital. All you need is power. That's all you need, an internet connection, right? Because the amount of fluids that you use is very small. It's just media. And the way that it's organized, you have a reference facility you can see on the top up here, right? And then you have manufacturing point of care facilities. And this is the model that we're going to be implementing here in Brazil, right? So actually 3 units are now going to be manufactured in the US. One is going to be deployed at Ohio State as the test unit, right, to be able to work out all the final protocols and everything, and 2 units are going to be deployed here in the Rio de Janeiro area. This is what one of the units looks like. I'm not going to spend a lot of time. Suffice to say is that up here you have manufacturing, and down here you have QC. So QC and manufacturing is all very close by, because what you don't want to do is you don't want to manufacture a product and then go to the other side of a hospital to do the test, right? You want to be able to do release testing real time, close, to make sure that you have a very consistent result. So access models and affordability. How are we different than others in the field, right? So if you look up there, this is your typical biotech pharma model, right? They make innovative products that displace current therapies. They have novel IP that protects a high price and market share. They are focused on value-based pricing models to justify the high price. They talk about, the whole package of cost in order that they can charge a high price. And they're motivated to justify the high price to keep full control of the product. And will only develop therapies that are fully controlled by the company and bring a rate of return that satisfies the shareholders. There's nothing wrong with this. This is the way that biotech works. And that's why you see biotech making choices for products that are profitable and not for those, for example, today we've seen with vaccines that are not profitable. This is the corporate world, and that's fine. Our model is a little bit different. We're a non-profit. We're focused on making just improved therapies, not novel. We're not so worried about IP, but we are focused on getting freedom to operate, right? So that there's no IP that prevents us from making these products. And we can do this based on our knowledgeable team. We know the patent landscape, and there's a lot already in the public domain. Gene therapy has been around for 20 years. A lot of the ideas have already been out there, and so all that is kind of expired. You know, it's already in the public domain. Right now, if you want to get a patent in the gene therapy space, it has to be very specific. It has to be either sequences or something. So if it's sequence specific, there's always a way to find around it to be able to create these products. We're focused on cost reduction, affordability, working with public health systems, for example, Medicare and Medicaid in the US. We're motivated to reduce the cost of the materials and democratize the process. And we're more aligned with hospitals and public health systems to really lower the price and broaden access. Oh, I'm sorry, I went back. And this is what we do, right, in a nutshell. On the left there, we're focused on manufacturing innovation, whether it's lentiviral vector technologies, which my team and I have been doing this for decades, cell manufacturing, the materials and reagents used in those processes, and then assay development. And on the left, you'll see some of our collaborators. Clinical candidates, leukemia, lymphoma, multiple myeloma, HIV, sickle cell, beta thalassemia, they're our top candidates. Some of our collaborators on the right over there. And then the logistical systems, the mobile clean room unit design, the point of care logistical systems of the networking, and then also AI data integration. And there are some of our collaborators there. So we're very focused on developing a robust, very simple process. I'm going to show you the process in a few slides here, but really taking a product, whether it's apheresis or blood, and that's an important discrimination because blood is a lot lower cost than taking apheresis. Selection, transduction. Now, for obtaining vector, we couldn't rely on a third party to make a vector for us that was cheap, right, or affordable, because they would charge us a lot. So we know how to make vectors, so we set up a company called Vectra Biomed. I'm going to share a couple of slides on that company with you. Then cell expansion, the candidates themselves, and then the logistics. And this is kind of our model, overall model, that you have a set of reagents, you have a vector, you have specific testing reagents, and that's distributed to centers that have a similar platform worldwide. They have the same instrumentation, and they have the same basic kind of reagents, like the medium, PCR equipment, and so forth. So that's our model, to keep things consistent throughout the network in the world. And this is some slides on our partnership with Fiocruz and Bio-Manguinhos. We're very excited by that. And here we are technology transferring everything we know. So we can build local capacity here in Brazil, not only for Brazil, but also for Latin America as well. So we're transferring both the CAR T-cell technology and the lentiviral vector technology. The cost will be about $35,000 per dose, not $375,000 per dose, and it'll be made available to the public health system without significant cost to the patients. The technology transfer is exclusive, not only for Brazil, but for all of Latin America. So we hope to be able to create an innovation hub here. And Fiocruz and Bio-Manguinhos will serve as a reference facility for clinical sites in Brazil and in other countries in the region. And we're envisaging not just a couple of products, but a pipeline of products here over the course of the next years. And I just want to acknowledge the vision and the support from the Minister of Health, who saw the importance of bringing these technologies to this country. And we're certainly very excited to be part of this collaboration. And the technology transfer has already started. A couple of weeks ago, some of the team members from Fiocruz and Bio-Manguinhos came to visit us, and we're already starting those initial first phases to be able to technology transfer the first part, which is the CAR T-cell manufacturing. And also, we just announced a couple of days ago that we're collaborating with a company called ImmunoAct in India. This is a company that did the very first CAR T approval in India. And so we're partnering with them to provide our TriCAR so they can bring it to patients in India and for commercialization. So very excited about that collaboration as well. And we're working very hard on a point-of-care network in the USA. The US, compared to many other countries of the world, has a very different healthcare system. You have a national healthcare system with the option of private payers, right? In the US, it's very fragmented. It's mostly private payers, and you have Medicare and Medicaid as kind of a safety net support for those that can't afford it. So the problem is many of those patients on Medicare and Medicaid don't get access, right? Because Medicaid and Medicaid, as you see there, will pay $257,958, I want to be precise here, for the cell product plus the patient care. Now, if the product costs $375,000, it's underwater, right? There's not enough money there to support the product and patient care. We believe that our material cost, as I'll show you, is $20,000. The lab and facility costs $10,000 to $20,000. So in the US, with the higher labor, we'll probably be in the $40,000 to $50,000 range to manufacture the product, leaving a price that you could be charged of about $100,000, leaving another $150,000 for all the clinical costs, which makes it viable in the US. So we are actively recruiting top centers at the moment in the US to develop this model. We're very excited by that. And just a couple of slides here on vector biomed. So we couldn't rely on somebody else making vector for us. So we said, we know how to make vectors. We're going to spin our own company. We're going to get some VC money around it, right? But we're also going to have a relationship with this company that it will make products for everybody else, actually about half price because of the efficient technology. But we'll have a special deal with Caring Cross to make these vectors for us at a much lower price so that we can work with others, to provide these vectors at a much lower cost. So I'm not going to go into great details. All that is that we're in the same facility with Vector Biomed in Gaithersburg here. Fantastic team. And I'm just going to show you the titers of the vectors. Normally you'll see titers of lentiviral vectors, final titers in the multiple 10 to the 8s. We get that in the harvest in the very beginning. So when you get such a high titer in the harvest, when you go to 10 to the 9, which is usually your target dose, you only have to concentrate about a 10-fold. That's really good because when you don't have to concentrate a lot, you then get a lot of aggregation, which means your yields are much higher. So we have a new technology here, and this technology will be transferred here to the Fiocruz team. And what the company does, it really provides speed, quality, and value for these products. And I want to spend one slide here on the economical manufacturing process that our Fiocruz colleagues were able to see just the last couple of weeks ago. So on the left there, you see we can start with whole blood or a leukopak. You have negative selection antibody that we've developed that you added in a single step. You get purified T cells, you put them in a G-Rex, add your vector, activation reagent, incubate for 7 days, put it back onto the Sativa Cepac-C Profa to formulate, and it's ready for infusion. Very inexpensive. And the material cost for this, including the vector, is about $20,000. And you get a very high cell recovery here, basically not only high T cell purity when you compare to positive selection, but also a higher level of gamma delta T cells. I'm running out of time here, but I'm just going to flick through the next slides very, very quickly. And basically what we have here is a very high percentage of stem cell memory cells. And we have a therapeutic pipeline of candidates. I won't have time to go into them in great detail, but we have a triple leukemia lymphoma CAR, HIV, sickle cell. These are our top candidates. As I mentioned to you, a triple CAR is better than a single CAR. These are animal experiments that show you why it's better. So these are animals that have received 4 different populations of cells to mimic relapse. If you look at here, the monocars, they get relapse. You have a triple CAR, no relapse, right? So here we have just some candidates, some very nice candidates showing good transduction efficiency, very good killing of the target cells. And this is some of the animal data here that we have here. You can see the CX170 here completely obliterates the tumor into the animals. And not only that, when we took these animals and re-challenged them again with the tumor, completely resistant again. So we're very excited about this product, taking it into the clinic, first in the US, and then we'll bring with some of that clinical data here in Brazil. HIV, I'm not going to go into it. There's not enough time. It's a duo CAR. We're in the clinic now. We're very excited to go to our next level of patient dosing. And for sickle cell and beta thalassemia, it's a very important disease, particularly in this country, especially in the Northeast here. But the price of the product is very, very expensive. And just to show you that this kind of product is curative. There's already a product on the market. It's a lentiglobin for sickle cell. You can see on the left there, the vaso-occlusive crises, that's all in purple. And then after therapy, no vaso-occlusive crises, right? So in beta thalassemia, right, you only need a single dose for sickle cell or beta thalassemia to be able to cure the patient for the disease. But the price is very expensive, $2.8 million. We're going to do it for much less than 1/10 of the cost here in Brazil. This is some of our preclinical data. It looks very good. We're very excited. And just one thing about our team, we're not just a people that have come into this field. We've been in this field. This is all that most of us have been doing for the last 20 or 30 years. So we have a lot of experience. We want to share that experience and make sure that as many patients as possible get access to these transformative therapies. And I won't go into the summary. I just want to thank our partners. This takes a village to do this. Actually takes a city to do this. And so partnerships for us are very, very important. We can't do this alone. Nobody can. And so that's why we're very excited with the collaboration that we have right here. Thank you very much. I appreciate your time.
Thank you, Boro. And now we have Martin Bonamino, who is going to talk to us about the use of those therapies in our public health system.
Okay, thank you, Antonio and Boro and Jim, who's not here. So it's kind of a tough topic to talk about gene therapy-based approach to treat patients in the SUS, because I think we have very few examples, if any, of this kind of therapy, especially for in vivo gene therapy. Most of the patients have been tried or have been treated now by using commercial products with prices such as those listed and mentioned by Boro and Jim. So this is a big challenge, actually. And I think the effort we're doing on several institutions here in Brazil aims to cover this kind of gap and try to offer this kind of therapy for most of the patients at an affordable price. So that disclosure made, and I think if we do this discussion again in 2-3 years from now, probably we'll have different things to say. But I will basically attach to the CAR-T model because I think this might be like a sandbox where some alternative has been tried and are under development, such as this, now under Bio-Manguinhos coordination on CAR-T cells and also for AAV-based vectors. But especially for CAR-T cells, we have a much richer environment to discuss the kind of approach we can take here in Brazil, right? So we'll be talking about mostly this right part of the complexity for cell therapy. So those will be genetically modified T cells. And just to remind all of you, we have more than 30,000 patients treated currently globally with CAR-T cell products. So very safe, very effective, very costly also. So this is what we have to tackle here, right? And of course, we are seeing this increase in complexity for these therapies. So Boro just mentioned that we're trying to keep this as simple as point of care and economically available or affordable approach. But the truth is that we are now seeing products that are derived from gene-edited cells, cells derived from iPSC cells, and we'll have like a myriad of different products being used here. So I think there will be room for many, if not all of these alternatives. But at this time, I think we have some examples for SUS based on more canonical CAR-T cells that are the autologous cells that are gene-modified with viral vectors or not, and then use it in autologous approaches for the patients. So I think this is no longer necessary to define the CAR molecule, but this is a chimeric molecule, and I think this is very important to highlight that this is kind of the crystallization of decades of development and knowledge on immunology and biotechnology. So it's a very beautiful example of what we can achieve once we're deciphering some biological mechanisms. And the other important thing here is that we have several flavors for CAR-T cells, so different modules such as the antigen binding domain, the signaling domain, and even the hinge and the structure of CAR-T cells can be changed. So as Boro just mentioned, we can design and redesign CARs and find ways of getting off the IP issue. So we can have freedom to operate if we design and combine different of these domains. So this is important because if we're about to develop new cars, we have to know how to do it and have freedom to operate to offer this to the Brazilian population. Right? So this is a short list of the products that have been approved. The 4 in red are also approved in Brazil. Most of them will have a tech price of about 2 million reais, which is clearly not reasonable for the public system. I think neither for the private system here. So all we have heard today is about how to do this in a more affordable way. So I'll try to condensate some of the concepts and solutions that we're seeing in the country, mostly for CAR T cells. But I'd just like to mention that for producing any of these advanced therapy products, we need like a change of goods and starting materials it's not available in Brazil yet. So from simple plasmids and DNA fragments to RNA, high quality RNA, and let go viral vectors. So we have some initiatives here. None of them is yet treating patients with virus or viral vectors produced in Brazil. So we think that in 2 years from now, 1 year from now, we'll have a new landscape. But this is the situation now. So we need desperately to have a change of production of goods and starting materials established in Brazil. And I think having big partners such as Fiocruz, Bio-Manguinhos, Butantan, and many others can be a good way to make this happen faster, right? So you have different ways of getting access to CAR T cells. One of them is ordering from big pharma. So I'll make an analogy here with the coffee, and you know this brand, but so you can say your name, they can make the cup of coffee, write your name, and deliver it to you. You can prepare on these automated machines, mostly like we do with coffee. You buy the capsules, you push the button, the machine will do it, all the process by itself. And you can also produce or develop. So here you seed the coffee, crop it. Maybe you can have some help from our Jacu friend here, and then have to collect them, wash them, right, roast, and then you can prepare your final coffee portion, right? If you put it in a perspective of production, you have like users, producers, and developers, such as you do for softwares and many other things. The same is true for CAR T cells. So now we have a very complex landscape in Brazil, many private centers using or buying CAR T cells. Some centers starting to produce them locally with automated machines or platforms, and some few groups developing things. So you just saw the presentation of a student from LOA's groups. There are many groups in Brazil developing things. None of them, or few of them, have the capacity to take it to the clinics. So closing the whole cycle of conception, development, and first in human testing is very challenging. So very few institutions can do this nowadays in Brazil. So if we can classify those centers, we'll have some producer centers, some of them using some automated platforms, such as the Prodigy platform from Minteni. So we have the Emocia at Ceará State, we have the Einstein Institute in São Paulo. We have some machines that have been already installed and deployed to Butantan, Ribeirão Preto. So those centers will produce or can produce now in these automated machines, but we have a very important bottleneck to getting these viral vectors, certified GMP compliant viral vectors to, and this is one of the goods that or starting material that you have, or one of the main materials that you put in your culture to transduce the cells that get them CAR positive. So some groups are teaming up with teams like Barcelona group that has developed an in-house manufacturer for the vectors. Some of them are chasing Bodo to get the vectors here in Brazil, and some of them are getting them from other centers. But this is a very complex situation where sometimes you buy the car, let's put it this way, but you have no fuel. You have some other developer centers, such as those that will use closed systems. So we have the example of the Einstein Institute, which have developed a manufactured protocol based on an automated system, and some others that use either automated and manual systems, such as the groups from Ribeirão Preto. And then you have some of the local point-of-care initiatives that we have heard now. So in the first moment for CAR T cells in Brazil, the patients were sent abroad. Then we have some clinical trials from industry, and then we had the approvals for these products and patients being treated in Brazil, but products that had been manufactured elsewhere. And now we have some academic clinical trials approved now, 2 of them in Brazil, one from the Einstein Institute and the other one from the USP Ribeirão Preto or Hemocentro de Ribeirão Preto, already recruiting and treating patients. So finally we get the local initiatives treating patients in Brazil. And we have many challenges and opportunities to develop these therapies and apply them and offer them to the SUDs, our health system. One challenge that we have an extensive network of blood banks and cord blood banks. This is a challenge in a certain way because now we have to decide what to do with these institutions, but on the other hand, we have several groups that have been trained now to do autologous and some of them allogeneic bone marrow transplantation. This might be the ground to apply very safely CAR T cell therapies now. We have a network of small labs that had been established a few years ago that were very important and are still important to try some proof of principle approach to new therapies that includes potentially CAR T cells. We have a huge country, so this is a huge challenge, how to offer this to several different regions of the country. We have the SUS, that's a big buyer and can negotiate prices. And we have also some viral vector supply challenge that hopefully some initiatives such as the one in Ribeirão Preto, the one in Bio-Manguinhos will start to taper by offering some of these vectors at least. And we have some opportunities. Many of them are the same on the list of the challenges because we can take this challenge and turn them into opportunities. So we have this capillarity of centers treating patients with cord blood, with transplants, and many other things. We have experimented centers for bone marrow transplantation that might be very important for managing CAR T cell toxicity and many other things. And of course, we have the capacity to produce some of these goods. So we have these huge, important institutions such as Bio-Manguinhos, Butantan, who have shown that they can engage and produce vaccines, biologicals, and now, why not, some of these viral vectors that we need. DNA, high-quality DNA, viral vectors, and so on. So one thing about CAR T cells is that they are just like any high-technology device where you can assemble and embed many technologies in. So you have the possibility of assembling CAR T cells in different ways that gives you some freedom to operate, that gives you some flexibility and a lot of room to improvements and evo designs. So this is something that we have been trying to do in our groups and many other groups have been trying to do in Brazil as well. We have now many CARs approved. Most of them will be against the CD19 target. Not the triple CAR that targets not only CD19 but also 20 and 22. But we have BCMA and CD19 as solo single targets being targeted by approved products in Brazil. And of course, if we want to innovate here, we have to design and validate new SCFVs and new antigen recognition domains. So this is why we set this initiative between INCA, Fiocruz, and also the Brasilia University that we call like the CAR T of Mucuripe. Mucuripe is a harbor in Fortaleza, right? So we have some of these representatives here. Giovanni is here, Marcos Lorenzon is here, and the UNB team where they have designed new SCFVs and we are validating them so we can have freedom to operate, to combine them in different ways, right? So we already have some new anti-CD20 SCFVs validated here, some anti-CD19 CARs validated here as well, and so they behave just the same as the benchmark FMC63 CAR. That's one of the approved CARs being used in commercially, or most of the commercially approved big pharma-based CAR. So here you can see that you can innovate, you can copy, you can do like a generic domain, and this is very important because then you have freedom to operate. Of course, they also work very nicely in vivo. And again, assuming that we have to try this, we should make like viral vectors, spend a lot of money, even if we want to try them in the first in human trial with very limited number of patients, like 8, 10 patients. So we're very committed to develop new ways here to push this to a first in human using like a fraction of the cost. That can be by providing viral vectors at lower cost. That can be by providing solutions that are non-viral vector based, such as the work Leonardo just showed to you all in the previous session, right? So the production models is also a very important topic here. So this all started in academia, went to very small labs, validating the first CAR products, then it was bought and acquired and incorporated by Big Pharma. And then not only Big Pharma, but everyone's trying to find and to develop consistency. And now some of these automated machines are making this much easier. But on the other hand, they are also making much easier to local centers that treat the patients to try to produce the CAR T cells at the point of care. So we're having like a movement of going back to decentralized production. And Bodo just showed an example of how it can be done. Some of these platforms are, so I'm not advertising here, so that's why I brought 3 of the main of them. But now we can expect to be flooded by different devices that can do pretty much the same, prepare the cells in different levels of automation, fully automated, semi-automated, and many other options. And then you can see that many groups will try to establish this in their hospitals and start preparing the CAR T cells using the materials. The other thing that's very important, as Bora just mentioned, is the viral vectors by itself or by themselves. So this can be very expensive, but also the viral vector and the CAR structure means that for each one of the CAR domains, you can use different molecules. And again, if you just combine these alternatives, you see that you can find several, maybe dozens of different CAR configurations and have freedom to operate in this design as well. Going back to the viral vectors, this is how much it costs in a facility abroad in the US or Europe. You can take more than 1 year to get a slot to produce your new CAR. So assuming that I have a brilliant idea, it will still take me like $1.2 million, more than 1 year until I get a GMP compliant viral vector. So this is not a good way to innovate, right? And it's also a full-time job to do this. It's not something that any amateur center can do. So that's where our production units, so I'm listing 2 of the units, but we have ST&I, Cimatec, Milena is here. We have other units that can play roles in producing this kind of vectors. So this is very important for us. So I'll just end up showing some of the models to approach the local CAR T cell production, right? So this is the industry model, centralize it. So you collect here, you send it to Netherlands or the US, depending on the company. And then you have to get your own cells, hopefully, not mixing up back. So you can use them in your patients in your center, right? So it's very complicated, as Boro just mentioned. You have a big slice of this cost that's dedicated to logistics and compliance and many other things. There's a local or regional approach on the development in São Paulo. That's the Ribeirão Preto initiative and the São Paulo Capital City initiative, where there's a very nice production center that has been established. Patients have to go there, cells will be prepared and they can be treated. It's a hub and spoke, but locally implemented. So it's very innovative, powerful initiative, but still patients have to go there. So you cannot imagine a patient from Acre and the patient's family moving to São Paulo for many months. So it's very complicated to get access to this in a large scale. São Paulo is a large state, so it's already a challenge to cover the São Paulo population by itself. And again, we have the initiatives that are not local in terms of this is the Carioca initiative we always play. So this is the car with a view. I bet you will not find a place with a better view to produce CAR T cells than this one. But this is a local initiative, not in the sense that we will cover only the local initiative or the local population, but this is a way of thinking, developing here the initiatives that are based in Rio, but they are developed already thinking about implementing this nationally, right? So let me just mention some of them. The non-viral CAR T cells that have just been shown by Leonardo. So this is the pipeline. This is a very cost-effective way of preparing the cells. We're planning to run a phase 1/2 trial at INCA with this initiative any moment in the next months. The manufacture will be held at the National Institute of Cardiology, where there's a certified clean room. So this is a joint initiative. Another initiative that is running now, it's the Children's Hospital of Philadelphia and INCA agreement, where we are establishing now a tech transfer with the very same protocol that has been used to treat the first pediatric patient. That's Emily Whitehead. Most of you probably know this story. And we're teaming up with this team, and they have been changing their manufacturers for an automated system. That's the Prodigy platform with their own lentiviral vector. They're transferring this, and we'll be treating about 30 patients with acute lymphoblastic leukemia, pediatric patients. So this is a way of speeding up things, and maybe, since this is a closed system, it could be an adapted way or an adaptation that we could take here and adapt most of these cord blood banks that are distributed across the country and that are not being used that much because, you know, the cord blood used for transplantation is going lower and lower, and so we have to think about how and what to do with these facilities. And last but not least, the Fiocruz Caring Cross / INCA also initiative that Boda just mentioned. And this is, again, a very interesting way because we're just teaming up two federal institutions that are located in the same place to try to validate this product and offer this later on to the SUS. So Boda has just shown the power of having this triple CAR design to avoid escapes from leukemia. There's another initiative that will be run in Bio-Manguinhos / INI, that's the Infectious Disease Institute here. That's a dual CAR for the GP120 target for HIV infected cells. But the thing here is that there will be this transfer of technology to produce the vectors. So this should make everything much easier. So having the containers is a very important step. This can be replicated in different places. So here we'll have a container at INCA and one container at Bio-Manguinhos to cover the Leukemia Lymphoma Initiative in INCA and HIV initiative in Bio-Manguinhos. And this model certainly is a hub spoke, but it can be replicated in a national level. So the idea here is that once we have this running here, several other states in Brazil can receive different containers with the same structure, the same quality control check, and compliance coordinated by Bio-Manguinhos, and why not to Latin America, as Boda has mentioned here, so it can go beyond Brazil. Right, so this is my last slide. Since I showed some of our initiatives, I'm just listing the people from my lab here. I'll just leave a huge acknowledgement to the institutions and funding agencies that had supported our efforts. And here I mean not only ours, but also others that we are developing with groups in Fiocruz and other institutions. So thank you very much.
Thank you, Martin, for exploring the many possible ways to bring those advanced therapies to our public health system. And now I think we have Marco Krieger online to discuss the presentations, and we won't have time for questions after Krieger's talk. Please, Marco, go ahead.
Sorry. I will try to do it in a very fast way. So first of all, I'm really pleased to be able to participate, even in a virtual way, in this important section of the International Symposium of Immunobiological, sponsored by Bio-Manguinhos. So I think the pioneers as far as of these different research that are all collaborating with Fiocruz in some different way in the field of advanced therapy, in addition to our production capability in Bio-Manguinhos, I think we will allow us to develop alternative strategies to guarantee access for our public health system patients to these new therapies. I think this is the important thing that we are trying to develop, and I would like to highlight the efforts of all teams that are involved in this project in Bio-Manguinhos and in Fiocruz, the legal team, the technical team that are working this project, as well as all the colleagues that are working this complex project. So I think in this panel we have Antonio Carlos that are working with us in the coordination of this project. We have Martin that is also belong to Fiocruz, but work also in Inca and have a very big contribution to our project and also to different initiatives. We have our international partners, Boro and Jim, that are all committed to the same project. The new developing of a strategy that will allow us to work in the local production of these of the vectors and the cells and the technology for the Brazilian public health system, but also bring it to Fiocruz Bio-Manguinhos an important role at the global level. So I think this is the message that we have here in this important section. So last but not the least, I would like to thank all the organizers of the Congress to allow me to participate in this session, even if I am in another meeting here. And that, I think, was really a very good section and a very good opportunity to show for our colleagues the Fiocruz initiatives in this field. So thanks a lot for this opportunity.
So unfortunately, as I mentioned before, we're running late, and I was told that we have no time for questions. But Boro and Martin will take any questions individually after we finish here, okay? Thank you very much.
Obrigada, Antônio Carlos, pela sua condução. Obrigada a
Thank you,
todos.
Antonio Carlos, for your moderation. To all participants, thank you guys. Again, don't forget the donations. You've seen that they can be made online. I don't know if you got the QR code. Do we have the QR code here? But please have access to the QR code. Please, if you can bring tomorrow your donations, water, non-perishable foodstuffs, cleaning items, and food for your pets, cats and dogs. Again, we'll see you tomorrow at 9 a.m. Again, thank you. Have a good evening and see you tomorrow. Thank you.